As we have already talked, that when amino acids are produced, they are also utilized, and there is a depletion of in the overall amino acid pool also in our body, in our cells. Similarly, the proteins also goes on being formed, and they also goes on being degraded. It is a continuous process. Both of these processes are important for the working of the proteins. You need proteins formation and the proteins, when they are in their intact form, they perform their functions. Very heterogeneous molecules they have. They are not only in terms of their structure, but also in terms of their function. So, in this way, proteins, first they are continuously being formed, second they are continuously being degraded also. That is a normal passage. Also, when they are being formed, there is also tendency of proteins to getting, a tendency of forming those proteins which are abnormal. Structurally, they are abnormal, so they can't perform their function. So, the system have to get rid of those proteins. So, in that way also the turnover of the proteins take place. There has to be a balance in healthy adults. The protein turnover should be in a way that there is a balance in the process of protein formation and protein degradation. Protein formation, of course, I have told you, gives you proteins. Protein degradation also is important because it also gives you the amino acids which are used then for all sorts of purposes. So, a normal individual turn over around 300 to 400 gram of body proteins on daily basis. That is a huge amount. Also, you should keep in mind that as far as the half life of the protein is concerned, their lifespan differs depending upon the which protein we are talking about, depending upon their different structural aspects. There are long lived proteins, there are very short lived proteins also. Protein degradation is a very complex process. It involves a whole range of different chemical pathways and also different protein molecules. In this pathway, that is the degradation of proteins, what actually happens that every protein which is going to be degraded, it is selectively degraded through a process known as protein ubiquitination. So, let me show you how this works. So, this is a protein, for instance, and then it is ubiquitinated because of the addition of ubiquitin molecules. They act as a signal for the protein to get degraded. It goes after ubiquitination. This ubiquitinated protein goes into a very complex structure known as proteasome. It is made up of a whole range of different protein molecules, and after a complex process of ubiquitination, and then the protein, the ubiquitinated protein enters into the proteasome, where it gets degraded, and it is divided into smaller molecules which are then further acted upon by different proteases, and we eventually come up with the finally amino acids, which are the main constituents of the proteins. So, this is one way of degradation of proteins, that is a proteasomal degradation, which is a targeted or selective degradation. Then there is also degradation which is non-targeted, which is general one, which is non-specific, that is lysosomal degradation. Is that the proteins goes to the lysosome, and then they are acted upon by the lysosomal enzyme, the hydrolases, which degrade the protein. The difference between proteasomal and this type of degradation is that in lysosomal degradation you have non-selective degradation, while in proteasomal degradation it is highly selective and highly targeted. Also, proteins, as I have already told you, have different half lives, and this half life depends upon the basis of their structural aspects. What amino acids is made up of, what is the arrangement of those molecules, amino acid molecules, and also it is very much influenced by specific N terminal residues and these specific N terminal residues give the signal for the degradation of the protein. Especially there are certain sequences of amino acids, for instance PEST sequence, which stands for the tetrad of amino acids, 4 amino acids in a sequence. This is P for proline, E for glutamate, S for serine, and T for threonine. These 4 amino acids are arranged in a way that they can give a signal for the rapid degradation of the proteins. So, in this way the presence of PEST is very significant in a protein. So, you see that how the turnover of the protein is regulated. The most important thing is that it is the very structure of the protein which determine when it has to be degraded and whether or not it should be degraded.